Thank you, Thomas, and thank you all for having me tonight. It's my privilege to tell you a little bit about what we've been doing from a research perspective in collaboration with some of my colleagues here, some of our local partners at McGill, nationally and internationally, to help better the lives of lung cancer patients both today and into the future. So lung cancer research has a very long history. In 1950, it was published in Life magazine, the link between smoking and lung cancer. And in 1964, the US Surgeon General issued a report stating that cigarette smoking was a very important health hazard in the United States and that action was required to reduce its harmful effects immediately. That was over 50 years ago. So we've been researching this disease and we've been looking for opportunities to help patients for well over 50 years. And as my colleague Jonathan Cools mentioned, we really haven't had many changes in survival outcomes in the last 20 to 30 years. But what I'll tell you today is that I think that's going to change, and I think that's going to change in the very near future, because we've had major advancements from a research perspective, and most of these things have happened over the past 5 to 10 years. So what's changed to allow us to get many strides in the last 5 to 10 years as opposed to the 50 years that we've been doing this kind of research? And to understand that, we have to understand that cancer is a genetic disease. So you just heard Dr. Fiset talk about how he gets a biopsy from the clinic, and he's able to identify within the tissue which cells are cancerous and which cells are normal. He can also take a look at those cells and say, this is a lung adenocarcinoma or a lung squamous cell carcinoma. But at the root of that cell, so what makes that cell look different is because it has an alteration or a mutation in its DNA or its genetic code. You see, all the cells in our body have the same genetic code. It reads kind of like a book. There's billions of letters in each cell in this code. And when something goes wrong, if you alter or mutate some of these letters or parts of this code, that's what ultimately leads to cancer. When you have these alterations, it changes the look of the cells and they become cancer, and that's what he's able to identify under the microscope. And so we know that smoking is one of the major things that can change or alter this code. So cancer is a genetic disease, and mutations lead to cancer. So if we really want to study or understand what are the root causes of cancer, we have to be able to read this code. And it's a very long and complex code. In 2001, we were able to finally sequence or learn the code of the entire human genome. Again, there's billions of letters in this code. And so the cost to do this in one single cell was over $15 million. So if I wanted to, as a cancer researcher, try and identify some of the mutations or alterations in a cancer cell, I would have to take a cancer cell from that patient. I would then also have to take a normal cell from that patient, sequence each of those for $15 million each, and after $30 million, I could say, well, he has an alteration in this specific gene that may or may not be associated with that cancer. So it really wasn't feasible just a short time ago. But with advancements in computing and in sequencing technology, we've been able to drive that cost down from over $15 million to less than $1,000. So we can sequence for a few $100s someone's entire genome. And the result of this is that over the last 10, 12 years, we've been able to sequence tens of thousands of cancers across over 30 different cancer types. And we've been able to probe what are these specific alterations which may be responsible for someone's individual cancer. In lung cancer, we've done this for over 1,000 tumors. And the result, again, just to reiterate, if Dr. Fiset looks under the microscope and diagnoses something as a lung adenocarcinoma, we've since been able to sequence and look at the genetic alterations in over 500 lung adenocarcinomas. This is publicly available data. This is from something called the Cancer Genome Atlas, which is an international collaborative effort to sequence as many cancers as possible and put that information in the public so anyone can go and look at those sequences and try and find out what are the genes responsible within those sequences. And from that, we sequence these tumors, and we've now started to develop a bit of a molecular portrait of these lung tumors. So for example, in this lung adenocarcinoma, we now know that there are certain alterations here that are the driver of these cancers. And with the identification of these genes or these alterations, we've been able to develop a number of drugs that target specifically these changes. I want to point out here, in this list of drugs that specifically target different alterations here, that only a few of them are actually approved or FDA approved. And so this is really happening in the last 5 to 10 years. We've been able to identify a lot of these alterations. There's been a big push from pharmaceutical companies to develop drugs that are likely to work against these individual cancers. And we're just starting to put these or implement these in the clinic. You just heard that standard of care here in Montreal is to a 15-gene panel for lung cancer. But they don't have that everywhere. That's not a national standard. That's really just a Montreal thing. In the United States, it depends which hospital you go to. They're starting to implement different panels for different cancers. But in the next few years, as it becomes more standard and we start seeing the fruits of this labor, this is going to become the norm for cancer treatment and diagnosis. And so another example of a lung cancer is lung squamous cell carcinoma, and we've again been able to identify a few of the molecular alterations which are responsible for this cancer and develop targeted therapies for these types as well. But I want to point your attention to these big, large gray boxes. We still don't know, even though we have the sequence, we still don't know what are the major drivers of nearly half of these tumors. And so that's something that we do in the lab. I do that in my lab right now, where we take these cancers, where we have no known drivers, so none of these alterations are found in these tumors or these alterations in these tumors, and we comb through these sequences and we look for alterations in other genes that are understudied. And then we find these alterations, we can take them into the laboratory, and we can test whether or not these genes may be the culprits that are responsible for these lung cancers. And when we identify these, we can then again work with pharma and try and develop inhibitors or drugs so that we'll have options for these patients in the very near future. So that's one of the things that we're working on in the lab. The second thing we're working on, which Dr. Fiset touched upon, is this idea of biomarkers of response. Dr. Cools introduced this idea of how lung cancer patients are treated today. And it's sort of a catchment, one size fits all. Of course, some patients that get diagnosed as a lung adenocarcinoma versus a lung squamous cell carcinoma versus a non-small cell lung cancer by a pathological diagnostic will get treated a little bit differently in terms of some will get surgery, some will get radiation, some will get chemotherapy, some will get combination of those. But in reality, they're going to get some combination of these therapies. Some patients are going to respond and have a very favorable outcome. Some will have no response at all. And there's actually a large group of patients that not only do not respond to the therapy that they're given, but they're succumb to the very harsh side effects. Some of these surgeries, as you saw, can be very invasive. Chemotherapy often can be very invasive. And so what we're working on in the lab is to try and develop and study new biomarkers. So we use all of this genetic information and we try and identify ways to identify before treatment which cancers are most likely to respond to a given therapy. So this could be implemented in terms of a blood test, a urine, DNA, or a tissue test. And so before treatment, we can identify those patients that are likely to respond. We can triage those patients onto the standard of care or what we're giving, whatever treatment ailment that we've decided for those patients. And then the other patients which we don't know, such as the ones that are likely to have adverse effects, or the patients are likely to have no effect whatsoever, we can then move those patients onto clinical trials to give them a chance. You see, we have so many drugs that are currently being developed, and we need patients to test these drugs. And I think this is something that we're really keen on developing, these biomarkers, so patients that are likely to respond can get the treatments that will help them, and other patients will have some options for their lung cancer treatment. And so just to summarize some of the things that we've been working on, of course, we'd like to identify before treatment therapeutic options for patients that are most likely to work for their particular cancer, using these personalized medicine approaches, that is to sequence their specific tumor and look for the treatment options that is most likely to be effective against their individual cancer. We're also looking to find new therapeutic targets in cancer patients that do not have any of the low-hanging fruit or the ones that we've been able to identify so far. And of course, all the time we're looking to understand the biology underlying these tumors, because there's a lot of similarities between many different cancer types, and some of the discoveries we find in lung cancer may also be beneficial for other cancers as well. And so the last thing I wanted to impress upon is that traditionally, research has been done in sort of a siloed manner in terms of, us scientists, like the pathologist, get stuck in the basement in our labs, and we're sort of like hermits, and that people think of us as introverted sort of lab nerds, and we do our thing, and then when we get a discovery, we sort of pass that on to the powers that be, and that eventually develops into a treatment option, which again can take decades. Our model in the Lung Cancer Network is that we work side by side with clinicians, clinician scientists, and researchers to work on and develop all of the projects, to biobank all of the samples, so we get accurate diagnosis from the patients, we follow the clinical information, and so this really is a team approach. And I think this is how we're changing how both research and clinical management of patients is going to be happening in the future by this bilateral relationship between the lab and the clinic. So with that, I want to thank you for your attention, and I look forward to any of your questions during the question and answer period. Thank you.